My name is Katie, and today I'll be presenting a paper called Polarity of Varicosity in Central Neuron Mechanosensation. So this paper talks about what happens to the brain when an injury occurs. And I have a personal connection with this because when I was 10 months old, my mother had a stroke, which left her wheelchair bound. And so that has led to me having a real interest in the brain and what happens to the brain when it's injured. And so I thought this paper tied in really nicely with a lot of my interests. So what exactly is a traumatic brain injury? It's generally defined as a disruption in brain function due to a blow to the head or a penetrating head injury. And usually when we talk about TBIs, we talk about two different stages. The first stage is the result of the actual physical force of the injury, and we usually see an influx of calcium into the neurons. We're going to be talking about this stage a lot in the paper. There's also a second stage, which involves excitotoxicity due to excess glutamate in the neurons, and this usually causes an inflammation of the dendrites. We're going to be talking about two main parts of the neuron in this paper, the dendrites and the axon. The dendrites are those branching bits coming off from the nucleus, and those are the parts of the cell that receive messages from other neurons. Then we have this long axon that usually is involved in sending messages, and these axons can either be myelinated or unmyelinated. This paper is all about a discovery that happened on accident. These researchers were doing a drug profusion experiment using Hanks buffer, which is basically a buffer solution of a bunch of different types of ions. And they realized that when they were puffing the buffer into the neurons, that these varicosities would form, and varicosities are areas where there is swelling in the axon of the neuron. So this really interested them, and they started to look into it further. So the first thing they did was try and see if there was a difference between myelinated and unmyelinated axons, and they found that there was.
Can you pause for me?
Figure A, you can
Because
see
I'm
that
just
on the top.
I'm a little slow. Can you describe varicosity to me again and exactly what that is?
So it's basically.
Oops, hold on, sorry.
That's basically how it's defined. It's just swelling. So like, you can see it even in the pictures, like in figure A, the bottom has those little like bumps that are sort of swollen. That's what the varicosity is.
Okay, so it's like these little tubers.
Yeah.
These little root vegetables. I see.
And it's gonna go into like how exactly that swelling occurs later.
So this is mechanical stress is inducing this. So how are they stressing these?
So they're using Hanks buffer, and they're like puffing it into the neurons, and it's that the pressure from the puff that's causing the mechanical stress.
So they're just adding in, and the Hanks buffer is not, it's just like kind of good for neurons. Is that?
Yeah, well, they were doing a whole other experiment, and they were doing a drug profusion experiment, and so they were using Hanks buffer in that experiment, and they just found that when they were using this buffer, these varicosities started to form, and so they were like, I wonder why that is, and they started doing more research into it.
So are they injecting it into the cells or just like into the media and like perfusing it over the cells?
Injecting it into the cells.
Okay, so like into the cell body?
Yeah. That's when they first started noticing it.
So they're doing tiny little micro injections like with joysticks, and that's,
and then later on do some like patch clamp stuff too.
Okay. Yeah. That's an interesting way. Sorry. That's an interesting way to increase like membrane tension or forces within cells that I've not really thought about, just injecting them with liquid.
Yeah.
Making them swell that way. I've definitely like played with osmolarity and stuff like that, so that's interesting. I like that.
Varicosities did not form in the myelinated axons, but on the bottom, you can clearly see that in the unmyelinated axons, these little bumps are occurring in the axons, and that's where the varicosity is, that's where the axon is swelling. They did not have a figure for this, but they mentioned in the paper that they wanted to measure the amount of pressure that needed to be puffed in order to cause a varicosity. So they used microfabricated silicone membranes, and they found the pressure that was needed to cause a varicosity to be about 190 millimeters of water. So they started looking at different parts of the neuron, and in C, D, and E, they found that varicosities.
What?
190 milliliters?
Millimeters.
Millimeters. Okay. Okay, I was about to say, how big is the cell?
No. Millimeters.
Okay, so they have like a small capillary tube and they're measuring like how many millimeters it takes?
Well,
That's
so
probably what it is.
I think they're trying to, like, you know how they measure pressure in millimeters of mercury a lot?
Oh, okay.
Like that, but they're using water instead of mercury, which I'm not quite sure why, but yeah.
Okay. Okay, so it's like it is a pressure then that they're actually getting out. Okay.
In distal and middle axons, but not in proximal axons or.
I'm gonna rewind that a little bit.
So they started looking at different parts of the neuron, and in C, D, and E, they found that varicosity is formed in distal and middle axons, but not in proximal axons or in the dendrites. They further found that more varicosities were forming in younger neurons as opposed to older neurons. They became interested in how this effect might occur in injured brains, and so they simulated several subconcussive impacts by puffing in different interval amounts. So they puffed for 2 seconds, and then they had an interval of differing numbers of seconds, and they found that varicosity was much larger at shorter intervals of time and not as large for intervals over 20 seconds. In fact, over 20 seconds, there was almost no effect. So
I don't like knowing this. I feel like now I know how to give someone a TBI really good, and it's repeated wax fast.
Definitely not fun.
So they used mice for this, and they gave them a traumatic brain injury by puncturing their skulls. They did it where that little black arrow is in figure A, and then they looked at their hippocampal neurons, and they found that in mice that had experienced the traumatic brain injury, many more varicosities formed. Given their previous data on the amount of pressure required to cause a varicosity, as well as the traumatic brain injury data, they thought that varicosities might be forming because of a mechanosensitive channel. So they went looking for the mechanosensitive channels that might be responsible for this change. To do this, they started removing ions from Hank's buffer to see which ions were responsible for varicosity formation. And they found that when they removed calcium and to a lesser extent sodium, varicosities formed less, and they were also smaller in size. This helped them narrow it down to 2 possible calcium channels that are responsible for this effect, the TRPV4 channel and the TRPV1 channel. They did whole cell voltage clamp recordings, as you can see in figure C, and found that the agonists for both of these channels activated them at normal levels, but the pressure from puffing only activated the TRPV4 channel, which made it the most likely candidate responsible for the varicosity formation. They used an antibody to determine that TRPV4 was expressed in hippocampal neurons, and it was expressed more in young neurons as opposed to older neurons, which makes sense with the data that they found in figure 1. They also used calcium fluorescence to determine that distal and middle axons had higher levels of endogenous TRVP4 channels, and they were the ones that had the largest amount of varicosities develop. They used a TRPV4 siRNA to partially knock down these channels, and you can see from the bottom figures that when they did that, varicosities were smaller in size and took longer to form. The researchers were very interested in why mechanical stress was causing varicosities in axons but not in dendrites. The system for glutamate excitotoxicity was already very well known, and it had been determined that glutamate excitotoxicity causes varicosities in dendrites, and they didn't know how it affected axons. So they decided to compare these 2 different systems. They found that excess glutamate affected dendrites like was expected, but it didn't do anything to axons. You can see this in figure B, where no varicosities form at all. They knew that glutamate in the dendrites affected microtubules, so they were wondering if this was something similar to what was happening in the axons. So they treated some axons with a microtubule depolymerizer and some different axons with an actin depolymerizer, and they found that only the microtubule depolymerizer had an effect and caused varicosities, as you can see in figures C and D. Previous research had shown that calcium disrupted microtubule formation by binding to the C terminus of the microtubule and destabilizing it. They used a calcium indicator to look at the amount of calcium in axons and in dendrites. So in B and G, we see these beautiful chymographs that are looking at axons and dendrites. In the axons, which is B, you can see that there's a lot of calcium for a sustained amount of time, whereas in G, there's sort of one point where there's a lot of calcium and then it goes away for the rest of the time. That data is quantified in the graphs below, which is just showing the same thing, that in varicosity spots, there is a very large amount of calcium for a long time in the axons, but there's only one spike of calcium in the dendrites, and then it goes low for the rest of the time. The researchers wondered how microtubule binding proteins affected varicosities, because usually microtubule binding proteins regulate calcium binding to microtubules. So they looked at 3 different microtubule binding proteins, and they found that the only one that actually had an effect at mediating varicosities forming was this protein called STOP. You can see in figure D that when STOP was present as compared to the other types of proteins they were looking at, there were much fewer varicosities. In either showing that as neurons get older, there is more STOP in them, which makes sense because earlier we saw that younger neurons were more susceptible to varicosity formation, and the fact that they don't have as much STOP is one of the reasons why this is true. In figure F, they also showed that STOP and TRBP4 channels were actually forming differentially. So you can see that there is more STOP in proximal axons, which is why those were less susceptible to varicosities, and there are more TRBP4 channels in distal neurons, which is why those developed more varicosities. They further solidified their data by doing a stop, knockdown, and then a rescue experiment. So when they knocked down stop, they found that many more varicosities formed, and they formed faster, and they were larger in size. However, when they added stop back in, they were able to rescue the cells, and varicosities formed less and also took longer and were smaller in size. Because the varicosities were affecting microtubule structure, they decided to look at microtubule transport to see if this was affected at all. Because one of the main things that neurons do is they are moving these different neurotransmitters to try and send signals, so this is obviously a very large part of what these neurons are doing. So they looked at different microtubule transport proteins, and they looked at them before they puffed in the buffer solution and after they puffed in the buffer solution. And what they found was after they puffed in the buffer solution, these transport proteins were not as fast, they couldn't go as far, and they were traveling less frequently along the neuron. And as you can imagine, this can have a very large effect on the way that you think, the speed that you think, if your neurons are trying to send signals quickly. So this last experiment that they did is the coolest one, I think. It kind of blew my mind when I was reading about it, so I hope it is interesting to you. Yes.
I just want to make sure I understand this right. Can we just go back a little bit? We don't even need to play it. I just want to look at it. The last slide. Yeah. Okay. This is like really striking, and these are cool looking chymographs of axon stuff, transportation. So chymographs are really often used for like microtubule transit, stuff like this. And so you can see we got these nice like slanted, huge tracks. So these like 2 red arrows, you can kind of follow those red arrows on these diagonal slants, right? And those track like one single train car that's going along the microtubule that's being pulled along. Sorry, these are just growth ends of microtubules. Oh, I'm sorry, I thought these were transport. So these are just, I can't hear you.
Oh, sorry, yeah, I was about to say, I don't think they're chymographs, but yeah.
Well, they are of microtubules, right? Because they're over time, yeah, but they're not, sorry, I thought they were looking at like transportation, like kinesins and things like that, but they're looking at end binding proteins.
Yeah.
So they're looking at polymerization of microtubules. Is that correct?
Yes, I believe so. Well, they're looking at like how quickly like the transport proteins are moving along the microtubule.
Okay, well, EB1's not a transport protein that I know of. It's an end binding protein. That's what it stands for. It doesn't really matter. We don't need to get lost in those details. The data is very striking. It does, if it's transport or end binding, it doesn't matter because it's microtubules that are important for both. And the data is just really striking. So they puff it and then they basically just stop moving almost. A lot of them do. It's crazy. Like these, especially in C, like look at how, like they're almost going sideways across that chymograph and then they just like all go straight. That's pretty nuts. Cool.
Neuron. And as you experiment that they did is so this last experiment that they did is the coolest one, I think. It kind of blew my mind when I was reading about it, so I hope it is interesting to you too. In this experiment, they used a patch clamp to measure action potential in neurons, and they were wondering whether or not these cells could tell that they had been injured. So they looked at action potentials before and after puffing the buffer solution, and what they found was action potentials actually traveled backwards after puffing the buffer solution. The researchers speculated that this was a way to inform the nucleus that something is wrong with the neuron, and they found that this action potential was actually induced more quickly in older neurons as compared to younger neurons. Finally, they had a model of this system. You can see the TRPV4 channels open under mechanical stress, allowing calcium to come in. They bind to stop. They bind to microtubules, causing them to disassemble. This can cause varicosities, and under prolonged mechanical stress, this can actually be irreversible. Whereas with glutamate excitotoxicity, that's only affecting the dendrites, not the axons, and this doesn't always cause cell death. This is my model. This big red structure is our axon, and the little structure on top represents a TRVP4 channel. At the top of the channel, there's a small trapdoor that can allow calcium to go into the axon. Inside the axon, we have our little creeper stop protein, and he is bound to the microtubule fibers in pink. When there is mechanical stress, as is represented by pushing this button, which activates a piston, the creeper stop protein is deactivated, which causes the neuron to make a varicosity in the axon, as you can see here.
Yeah.
So can we watch that model again?
Yeah.
That first time was just the tease. We got now I really want to absorb this again.
My model, this big red structure is our axon, and the little structure on top represents a TRVP4 channel. At the top of the channel, there's a small trapdoor that can allow calcium to go into the axon. Inside the axon, we have our little creeper stop protein, and he is bound to the microtubule fibers in pink. When there is mechanical stress, as is represented by pushing this button, which activates a piston, the creeper stop protein is deactivated, which causes the neuron to make a varicosity in the axon, as you can see here.
It's so okay. That's so interesting. Cool.
Yeah, okay, so I have a discussion assignment. So if we could use the whiteboard app thing.
Yes, we can. I actually have it open. Or do you want to do it? Or I have it open.
If you have it open, that's cool.
Yeah, I just gotta remember how to share the link because I never remember how to do this. Add people. There we go. Copy link. And link is in the chat.
Okay, so I basically just wanted to see like what everybody thought was interesting about this paper and maybe what they learned. And so I basically just want you to draw out like what is the coolest thing that you learned about neurons and varicosities from this paper. I drew out my little picture, it's on the slide. I drew the action potential going backwards when there's varicosities, and the neuron on the bottom is like, oh my gosh, we're injured. So I basically just wanted to see what everyone thought was cool and if people wanted to share about that. And then the second question is basically just how do you think that this research might inform biopsychological medicine and can inform clinical practice, essentially. I can go back to their model too if you need to be reminded about what exactly happened.
Yeah, that would be good. You can be on the model for a little bit, and then you can screen share your board. You can just go to that board, the whiteboard, and then you can screen share that after a while.
For sure.
Yeah, I mean, as we're drawing, I'll just talk about my thing because I want to talk about it because I didn't know any of this, and it's really kind of wild. I'm really into. I want to look at like the histology of these and look at like the myelin sheath and see what's happening with that too. Because it just seems like these various varicosities, they're interacting with the outside cells, the myelin sheath probably, and also the internal cytoskeleton is affecting them obviously. Through the microtubules breaking down and all that, like they talked about. So yeah, I kind of want to just know more about these little structures and like kind of the rules that when we break this little part of it, why do the rules that are in place cause this to happen? I'm interested in these varicosity structures and why they're forming.
For sure.
Yeah, that's interesting. I want to like cut up neurons and do EM on them and look at all those. I want to know what's going on in these structures at the like molecular level and what their architecture is and stuff like that. It's very cool.
Okay.
And so I actually had another question too before you start the class discussion. So early on you said that they were injecting it into the cells, but then in that later figure they were perfusing it over the cells. And so so was that they were switching between the two?
Yeah. So in the beginning, because they were doing that other experiment, what they were originally doing was injecting it into the cells. But then once they started doing their data for like this specific experiment, they were putting it outside. Yeah.
And but it produced a similar effect.
Mm-hmm. Yeah.
So then how do they know it's the pressure? Does that make sense? And not just like the media?
Well, because they mentioned early on that they like had varied the amount of pressure and it took like a certain amount
of
pressure in order to make
there's a threshold. So they could loose limp the media in there and it doesn't do this.
Yeah.
Okay, cool. All right. Did people get into the board?
Yeah, there's a couple people here.
Okay. Oh yeah, I drew stuff. Mine's just really tiny.
Okay. I can start sharing that board if you want.
Yes.
All right. So does anyone want to talk about their drawing? I see some like cool backwards action potentials going on.
Yeah, can you just like cruise around the board for us?
Yeah. So
just kind of look at these. I like that. This says ouch.
Yeah.
Also like the arrows kind of like walking back slowly.
Yeah, me too. That's cool.
I can talk about mine for a second. Mine's the one that says ouch.
Okay.
I agree with you. I thought the coolest part was just the fact that like it can go backwards because I never really thought about that. So that's my little action potential going backwards. Since whenever I think of action potentials, I think of it going from like one channel to the next channel. And I always knew that once it passes a channel, it makes it so it can't go back to that channel that it just passed. But I never thought about the fact that it could go the other way as well.
Yeah, same. I like every class you ever talk about, they only go forward. And so I was like, oh my gosh, it totally makes sense that they could also go backwards.
Yeah, it's just something I never thought about.
Yeah, in general, I think we make electricity way more complicated for whatever reason that it is, but it's something that is always difficult for me to grasp too, of like which way, it's just like water though, it can flow both ways.
Anybody else want to talk about their pictures at all?
Yeah, I mean, I think people thought the paper was cool, so don't take that as the lack of discussion. Is that, you know, we are just over time already, and we have 2 more presentations. But I thought I'm grateful that you presented this and that you shared your kind of, you know, your own family's experience and stuff, and your experience in a fascinating way. So thank you for doing that.
Of course, yeah.
Did you have anything else you wanted to?
No, just like if people wanted to talk about the clinical applications, that's cool.
Yeah. Oh yeah, I forgot about that last little part. Let's do that.
So does anyone have any thoughts on that?
I could start. I was way into this idea of neuron development and neuron lifetime now, and how these older neurons have this protective armor called end stop. So it don't matter if you want to become so what this says to me is, if I want to become, let's be honest, I'm going to be a featherweight boxer. But I think 36, now that I'm older, it's a good time to start boxing, because I won't get a TBI as easily.
Yeah, there you go.
So if you want to be a boxer, wait.
Exactly.
Wait.
That's the moral of this paper.
No, but I think there's a lot of medical relevance in here.
Mm-hmm. Yeah, for sure. I think so. They were mentioning earlier on in the paper that a large amount of varicosities actually is like an identification factor for like Alzheimer's and different neurodegenerative diseases. Yeah. And so I thought thinking about that was really interesting, how mechanical stress might be playing into these other diseases that we might not even know about.
Yeah, did they look at all at the protein contents?
No.
Like, do they have like plaque like things forming in there, like density? I'm sure they do have like.
Yeah, they didn't really look at that at all in the paper. Honestly, the paper, some of it, I felt like they could have gone into a lot more detail and they didn't, and so yeah, I don't know.
Yeah, it's always a dance you got to do. How much do you put in? How much do you allow the reader to figure it out? Yeah. Cool. Okay.